Hi everyone, welcome back. Today we're going to be discussing primarily Chapter 15, but also a bit of Chapter 16, and we'll be discussing autoimmunity. So today we're going to start off just generally discussing what is autoimmunity. Then we'll be talking about distinguishing between peripheral and central breaks in tolerance. And we'll also learn the roles of both the innate and adaptive immune system. For example, autoantibody and T-cell involvement. Then we'll also talk about antigen cross-reactivity and molecular mimicry as mechanisms for autoimmune disease. And we'll touch a little bit on treatments for these different autoimmune disorders, but we'll really go over this in detail in Chapter 16, or the Biomodulators lecture. Finally, we'll wrap things up by discussing some of the triggers for autoimmune disease. Okay, so what is autoimmunity? Essentially, autoimmunity is a break in self-tolerance, where the immune system actually targets itself. So think back especially to the lectures about tolerance during T and B cell development. So we'll touch a little bit on some of these mechanisms, but they were covered in detail previously, so you can also go back to those lectures. So we're going to be specifically focusing on how the breakdown of these mechanisms of tolerance can lead to autoimmunity. So autoimmune diseases roughly separate into systemic autoimmunity, where we have autoreactivity against an antigen that is found throughout the body, and organ-specific autoimmunity, or a disease where the antigen that's targeted is found only in one organ. So here in figure 15.2, we have a list of several autoimmune diseases that we'll discuss today. And a couple interesting ones include Crohn's disease, which is actually a bit more complicated as it's really an autoimmune targeting not exactly of the self, but actually of the microbiota. And we'll also discuss the differences in systemic and organ-specific disease. So really, you don't need to memorize these diseases, just think of this for the concepts. So especially the different types of mechanisms that can lead to these diseases and how breakdowns in the immune system cause this. Okay, so the major mechanisms of autoimmunity include autoantibody, which can be against cell surface proteins and target them for destruction. Or they can also be against receptors, which disrupts the function. Another major mechanism is T cells that are self-reactive. And in addition, we'll be talking a bit about immune complex formation. So, as you guys have talked about in previous lectures in development, there are several different types of tolerance. So we'll be talking a bit more in detail, especially about central tolerance today. So self-tolerance in general is very important mechanisms by which the immune system learns not to attack itself or self-proteins in the body. So again, think back to T and B cell development, and especially in this example, think about the thymus. So this is where T cells develop, and we talked a lot earlier about mechanisms by which T cells that recognize self-proteins are eliminated, thereby reducing the risk of autoimmunity. Self-tolerance, though, is really just the first important checkpoint. So if we think about B cells developing in the bone marrow, we'll have one B cell that undergoes somatic hypermutation, creating many different clones. Then some of these actually may be able to bind self-antigen, and the encounter of B cells with self-antigen within germinal centers, if everything works correctly, should cause apoptosis of the self-reactive B cells. We also need to consider that some areas of the body are sequestered from the typical surveillance of the immune system. The most well-known privileged site is probably the brain. You guys have probably heard of the blood-brain barrier, and this helps keep out different cells of the immune system because it can lead to severe consequences if the immune system begins to attack the brain, or even if the immune system leads to inflammation after an infection in the brain. A couple other organs that are typically sequestered include the eye, the testes, and the uterine-fetal interface. So there are some really important positive feedback loops in autoimmunity. Self-antigen, due to the fact that it is from the self, is not easily eliminated. So this leads to continuous inflammation and therefore recruitment of more and more effector immune cells. We will discuss this a bit later, but this can also lead to epitope spreading, which is an increase in the antigens that are recognized by the immune system. So new lymphocyte clones, as they undergo mutations, can react to new epitopes. And B cells can also present new autoantigens to T cells. Cryptic epitopes are revealed as internalized autoantigens, and so this would be particularly when they're presented on MHC molecules. So this is an example of some antigen spreading, some positive feedback loops here. So if we start off with one B cell, this circulating B cell may bind self-antigens released from an injured cell. This B cell then is activated by a T cell specific for this peptide, and B cells can then differentiate into plasma cells and secrete antibody. So then, if we have a site of injury, self-antigen specific antibody can initialize an inflammatory response. So this leads to more cell injury, thereby releasing more self-antigens, and then more self-antigens can be recognized by additional B cells. Another way that epitope spreading can happen is if we have a CD4 T cell that's specific for one epitope, then it can provide help to a B cell specific for other accessible epitopes of this complex. So this is figure 15.7, if you guys want to see it in a bit more detail. And so a B cell that can internalize different epitopes or proteins can then present those new peptides to T cells, which might recognize them. So we have kind of this positive feedback loop between these different cells. And during autoimmunity, multiple parts of the immune system are recruited. So if B cells are recruited, we may have autoantibodies that target specific antigens or can form damaging immune complexes. If T cells are involved, we may have effector T cells that can cause inflammation and also direct tissue damage. So typically we'll actually see a combination of these two factors. So this is just a table summarizing some of the different parts of the immune system that might be recruited. So here we have a couple examples of different autoimmune diseases. So, for example, in systemic lupus erythematosus, or SLE, we have T cells that are pathogenic and can provide help for antibody. We also have B cells that produce autoantibody and can also present antigen to T cells. Another example would be type 1 diabetes, which is an organ-specific autoimmune disease. Here, the B cells largely present antigen to T cells, and then CD8 positive T cells will enter the tissue and destroy it. So when we're looking at autoimmunity, often it's hard to tease apart which part of the immune system is causing the disease. So there are a few ways we can try to answer that question. The first is that we can transfer different components of the immune system, particularly antibody. So this can be done in a laboratory by transferring B cells or autoreactive T cells from one mouse to another, thereby transferring the disease. And this can also happen naturally during pregnancy. So for example, in a patient with Graves' disease, which involves many autoreactive antibodies, these antibodies can actually be transported across the placenta, and then the newborn infant, whether they have a genetic predisposition or not, will actually develop Graves' disease. In the few months after birth, these maternal antibodies will start to degrade, and then the infant will improve and no longer have the disease. So this is a natural way that we can figure out which parts of the immune system are leading to the disease. So transfer of components can establish disease causality, like I said. So some examples here in figure 15.13 involve Myasthenia Gravis, where we have an autoantibody against an anti-acetylcholine receptor. And in mice, you can actually transfer this antibody, thereby transferring the disease. So I've mentioned a few times the role of T cells. So effector T cells can actually cause tissue damage directly. And this is particularly seen in type 1 diabetes, where we see the destruction of beta cells in the pancreas. So here, T cells that are autoreactive for the beta cells, particularly CD8 killer T cells, will enter the tissue and destroy those beta cells, thereby destroying the ability of the individual to respond with insulin. And there are examples across other diseases of this showing. You guys can review this table on your own. We're not going to be going into detail about all of these, but it's a good way to think about what might be causing different diseases. So autoimmunity actually often reflects a dysregulated response to pathogens. So the immune response we typically think of as fighting off infections, and autoimmunity can actually occur as a result of this becoming dysregulated. So we see that type 2 immune responses usually don't play a role, but tissue damage by autoantibody is more likely linked to dysregulated Th17 or Th1 immune responses. We can also see T-independent IgM autoantibody that can cause tissue damage by the formation of immune complexes. So autoantibody against blood cells can lead to their destruction. So this is one example of how autoantibody can cause autoimmune disease. So in an individual, they may have IgM or IgG that binds to an antigen on the surface of a blood cell. And this antibody, just as we've talked about previously with antibody opsonization or complement, then targets these cells for destruction, again, either by phagocytosis or by the complement cascade. An example of this is autoimmune hemolytic anemia, which from the name we can see represented in this chart. The red blood cells are coated in antibody and then destroyed, and the reduction in red blood cells therefore leads to anemia in the patients. Lysis of nucleated cells, also via complement, is less common. So it's more common that your red blood cells, which lack a nucleus, will be targeted for this. So the development of anemia is more likely. So on the other hand, while they may not be targeted for complement, they may be destroyed via phagocytosis by NK cells or via ADCC. So complement is still destructive even at sublytic amounts of fixation. So even if you don't have enough complement protein to lead to lysis, for example, of red blood cells, it can still cause tissue damage. So complement deposition stimulates a general strong inflammatory response, including cytokine release and respiratory burst. Then immune cells will be attracted to the area because some complement proteins are chemoattractants, for example, C5a. Leukocytes might then be activated, and this can happen by binding autoantibody, the Fc receptor on cells, can bind the Fc region of antibodies, or the binding of complement protein C3. And this response can lead to tissue damage. Autoantibodies also can disrupt cell function. So, in particular, autoantibodies can block cell surface receptors, and they can either block them actually or stimulate them, and these can lead to different types of disease. And typically, in a disease where this is an important part of the mechanism, the blockage of these functions will actually lead to the symptoms of the disease. So a good example is Graves' disease, where autoantibody is produced against the TSH receptor, and this leads to an increase of thyroid hormone. Another very well-known disease where this occurs is in Myasthenia Gravis. So in some types of Myasthenia Gravis, we have autoantibody that's produced against the nicotinic acetylcholine receptor. And this leads to internalization and degradation of the receptor on the surface of cells, and thereby leads to muscle paralysis. So here in this figure 1522, we can see in a normal neuromuscular junction, we have the neuron, which is shown at the top in gray, can release acetylcholine, and a muscle cell will then see this acetylcholine via the acetylcholine receptors, and this will lead to muscle contraction. However, in Myasthenia Gravis, we have autoantibodies that come in and block this receptor, and this then leads to internalization of the receptors, and the muscle cells can no longer respond to acetylcholine release by the neurons. So this leads to progressive unresponsive muscles that no longer contract. So additional autoantibodies that are against extracellular antigens can cause inflammatory injury. So this is an example of how this damage can happen in the kidney. So the basement membrane is really an important barrier between blood vessels and other parts of tissues in the body. But antibodies that bind to the basement membrane in the kidney will, again, like we've talked about, activate complement and recruit immune cells like neutrophils. And this leads to destruction of cells in the kidney, as well as you can see here. This is a glomerulus, and this area here is significantly thicker than it should be in a healthy patient due to this enormous influx of inflammatory cells. So a few examples here of diseases caused by this mechanism include, like we talked about, autoimmune hemolytic anemia, as well as pemphigus vulgaris, or acute rheumatic fever, which we'll touch on a bit later. So I've mentioned a few times damage that's caused by immune complexes. So what is an immune complex? An immune complex is this structure that forms when an antibody responds to a soluble antigen. And in this case, the antibody will cross-link the antigen and form larger and larger structures as multiple antibodies and antigens come together.